All right, well, moving on to 23.4, about leaves. The goal is a little bit different this time, get rid of that, is to relate structure and function for the components of plant and animal cells, explain the role of cell membranes as a highly selective barrier, as you remember, passive and active transport too. Our two objectives are to describe the structures of a leaf and how it enables it to carry out photosynthesis, and explain how gas exchange in leaves relates to homeostasis. So you have heard a lot about being in green industry, green buildings like our high school building and everything like that. But did you know that the most important manufacturing sites on Earth are already green? And they are the leaves of plants. They use the energy captured in the leaves. Plants make sugars, starches, and oils that feed virtually all land animals, including us. So our first objective is to describe how the structure of leaf enables its leaves, enables it to carry out photosynthesis, or leaf. To collect sunlight, most leaves have a thin, flattened part called the blade, as you can see right there. The flat shape of leaf blade maximizes the amount of light it can absorb. The blade is attached to a stem by a thin stalk called a petiole. Leaves have an outer covering of dermal tissue and inner regions of ground and vascular tissue too. So let's look at that, the layer of a leaf. The top and bottom surfaces of a leaf are covered by the epidermis, which is a tough, irregularly shaped cells with thick outer walls. The epidermis in nearly all leaves is covered by a waxy cuticle, which is a waterproof barrier that protects and limits the loss of water through evaporation. Phloem and phloem tissues are gathered together into bundles called leaf veins, right here, that run from the stem throughout the leaf. They protect the leaf and limits water loss through evaporations. The upper epidermis is a layer of cells called the palisade mesophyll, containing closely packed cells that absorb light that enters the leaf. Beneath the palisade layer is a spongy mesophyll, right here, which has many air spaces between its cells. The air spaces in the spongy mesophyll connect with the exterior through stomatas, small openings in the epidermis that allow carbon dioxide, water, and oxygen to diffuse into and out of the leaf. The walls of the mesophyll cells are kept moist so that gases can enter and leave these cells easily. However, water also evaporates from the surfaces and is lost to the atmosphere. Transpiration is a loss of water through leaves. This loss of water may be replaced by water drawn into the leaf through the xylem vessels in the vascular tissues. Transpiration helps to cool leaves on hot days, but it also may threaten the leaf's survival if water is scarce, and as this is seen in a wilting plant. So our objective was to describe the structure of leaves and how it enables it to carry out photosynthesis. The structure of the leaf is optimized to absorb light and carry out photosynthesis. Next objective is to explain how gas exchange in leaves relates to homeostasis. So leaves take in carbon dioxide and give off oxygen during photosynthesis. When plant cells use the food they make, the cells respire, taking in oxygen and giving off carbon dioxide. Plant leaves allow a gas exchange between air spaces in the spongy mesophyll and the exterior by opening their stomata. If a stomata were kept open all the time, water loss due to transpiration would be so great that few plants would be able to take in enough water to survive. Plants maintain homeostasis by keeping their stomata open just enough to allow photosynthesis to take place, but not so much that they lose an excessive amount of water. So they have guard cells. Guard cells, as you see in this figure here, are highly specialized cells that surround the stomata and control their opening and closing. Guard cells regulate the movement of gases into and out of the leaf tissues. Carbon dioxide can enter through the open stomata, and water loss is lost by transpiration. When water is abundant, it flows into the leaf, raising the water pressure in the guard cells, which opens the stomata, as so. The thin outer walls of the guard cells are forced into a curved shape, which pulls the thick inner walls away from one another, opening the stomata. When water is scarce, though, I should have clicked over, sorry. When water is scarce, water pressure within the cell then does decrease. The water inner walls pull together and then the stomata closes. This reduces further water losses by limiting transpiration. In general, stomatas are open during the daytime when photosynthesis is active and closed at night when open stomata would only lead to water loss. However, stomatas may be closed even in bright sunlight during hot, dry conditions in which water conservation is a matter of life and death. Guard cells can respond to conditions in the environment such as wind and temperature, helping to maintain the homeostasis within a leaf. Osmotic pressure keeps the plant's leaves and stems rigid. High transpiration rates can lead to wilting. Wilting results from the loss of water, and therefore pressure in the plant cells. The leaves of these plants have adaptations to the dry or low nutrient conditions in which they live. And there's several we're going to talk about. The first one is called the pitcher plant. The leaf of the pitcher plant is modified to attract and then digest insects and other small prey. Such plants typically live in nutrient-poor soils and rely on animal prey as their source of nitrogen. I missed this one. This is called the rock plant, and it simulates looking like a rock. It has lots of exposure, the leaves, lots of exposure, surface area. I gotta tell you more about that when I find it. The spruce, it has narrow leaves of a spruce contain waxy epidermis as well as stomata that are sunken below the surface of the leaf. These adaptations, waxy and stuff, help reduce water loss from the leaves. Cactus leaves are actually non-photosynthetic thorns. The thorns are considered the leaves that protect them against herbivores. Most of the plant's photosynthesis is actually carried out in the stems, which we learned about before. So objective 2 is explain how gas exchange in leaves relates to homeostasis. Well, plants maintain homeostasis by keeping their stomata open just enough to allow photosynthesis to take place, but not so much that they lose an excessive amount of water. So our two objectives describe how the structure of leaves enabled to carry out photosynthesis and also explain how gas exchange in leaves relates to homeostasis. And they both help us to relate to our goal for this lesson. And there's a cute little picture of a kitty.